Okay, hello everybody. Today we're going to be presenting a paper called DsrA RNA Regulates Translation of the rpos message by an anti-antisense mechanism, and this is independent of its action as an anti-silencer of transcription. So that sounds like a lot, but I promise in the next few minutes you're going to understand what that means. This paper is pretty dense, but hopefully by the end of it, you're going to have a very good understanding of it. So, what we're going to go over today, we're just going to take a quick look at the E. coli genome. We're going to look at the functions of DsrA RNA. We're going to look at the experiments and the methods that the authors used regarding DsrA RNA. And our main focus today is going to be on the first stem loop of the DsrA RNA. So this is just a quick map of the E. coli genome. I just put it in here because I thought it was interesting. These black bits here are the non-structured regions. These are the various domains of the genome. And just to show you what it actually looks like, this is how it's coiled and super coiled up in order to fit within a bacterial cell. So that's just a little aside. Now let's get to the main content. So the star of our presentation today is the DsrA RNA. It has 3 stem loops, each with a distinct function. Now on the left you'll see the way our authors depict it, and on the right is just another picture I found, just a little alternate prediction. So the functions of the DsrA RNA, and the authors knew a little bit of this going into this paper. So our first loop is responsible for promoting the translation of the rpos protein, and this is a stress factor found in several bacteria. Now, our loop 2, the purpose of this is to overcome the silencing which is caused by a separate protein, HNS, and we're not going to dig into that too much. We'll just go over it briefly. And loop 3 is a row-independent terminator. This is something we've studied in the past, pretty much. As we're creating the DsrA RNA, that just says transcription's done, the DsrA is done. Let's get it out there. So just looking at how loop 2 functions, just a little bit of background info you're going to need. The H-NS protein is responsible for preventing the expression of the RCSA protein, and the RCSA protein is responsible for promoting the expression of the CPS genes. These are the genes which encode the capsule. So what our DsrA RNA does, and specifically this function can be attributed to the second loop, it will counteract the action of H-NS, and it will allow RCSA to activate those capsule genes. This is something that occurs when DsrA RNA is overproduced, and we're not really going to dig into the mechanisms behind this in today's presentation. We're mainly going to focus on loop 1 and go over our experimental setup. Now, our E. coli that we use in these experiments are going to have their natural lac operon deleted, and they're going to have their natural DsrA deleted. So what we're going to do is we're going to take that lac operon and we're going to fuse it over here to the rpos gene. So we're also going to insert a plasmid into our E. coli, and that is going to have DsrA under the control of the PBAD promoter. Now pretty much, I'm not going to go into too much detail, but we just use the PBAD promoter so that this sugar down here, L-arabinose, is going to be used to activate DsrA RNA production. We've given ourselves just an easy way to turn it on and off by applying different amounts of that sugar. When we look at figure 2, what we're really looking at is just testing that little experimental setup that we have. So when you look at this first chart over here, The units that we're measuring in are in specific activity, which is that's just a measurement of enzyme activity. Down here you can see the percentage of arabinose that we grew the E. coli in, and this right here just denotes that that percentage of arabinose creates a similar activity as the wild type version would. Now these here vectors, these are just controls. They don't have DsrA. And over on the right, we're just looking at the decay time, trying to get to the half-life of some different RNAs. So if you look, our DsrA is very stable. This DsRB is less stable, but that's not too important. This over here just indicates a version of that plasmid that had multiple copies of DsrA. So the way that we got this chart over here is they just added this chemical to stop transcription and measured the amount of the DsrA RNA over time. So the way that we measure the amount of the rpos translation that is occurring, whenever rpos is translated, as you remember, it has that LAC-Z attached to it. So we just use an ordinary beta-galactosidase assay to measure that. We should all be pretty familiar with that by now. So what did we learn from Figure 2? Just a quick review. Our PBAD promoter setup that I showed you guys earlier is functional. We can use arabinose to kind of modulate expression of DsrA. You know, more arabinose when we grow the E. coli makes more DsrA. We know that our fusion of rpos to LAC-Z is working, and we know that our DsrA RNA doesn't really decay, which that's actually going to bring up some questions later on, but we'll get to that. So now let's go further into our experiments and look at Figure 3. So the way that we measured anti-HNS activity is with a CPS, and remember CPS is those capsule genes, and we fuse those to LAC-Z, and that's how we're going to test our loop 2, which is responsible for that activity. Now rpos translation, we're going to measure that with an rpos LAC-Z fusion, and loop 1, as well as this little area here, is mostly what's responsible for promoting the translation of rpos. So what's our goal here? Our goal is to just kind of map out the domains of the DsrA RNA and attribute the functions to each domain. Now we're going to need to go into restriction sites. Here's where we inserted a restriction site at one point, and here's where we inserted another one. Basically what that means, we took a little bit out and replaced it with this certain sequence, and these certain sequences serve as like a target for an enzyme to cut there. So we'll get more into that later on. Okay, so if you look here, we generated several different mutants of DsrA RNA that we're going to use in our experiments. I know this looks like a lot at first. Literally all we're measuring in this, we're using a northern blot, and we're just making sure that we have enough of each of these RNAs. Basically that transcription is able to occur in our mutants. So if we look at lane 1 and lane 2, this is just our ordinary setup, the PBAD promoter with different percentages of arabinose. Now our deletion of 1, let's just go ahead and go back and I'll show you what that means. It just means that we cut off stem loop 1. Our deletion of 2 just means that we cut off stem loop 2. So as you can see, we have some RNA in all of these, but when we delete stem loop 2, transcription doesn't occur very well. So normally there was 5 micrograms of RNA in each of these lanes. In this one, they increased the amount. So over here on number 6, What they've done is replace stem loop 3 with trip A. That's just a different transcription terminator. Pretty much does the same function as what loop 3 does here. Now when we look at lanes 7 and 8, that's just where we've inserted our restriction sites, and I'll just show you on the larger image. Here is the in-call restriction site, and here is the ex-haul restriction site. So that's what those refer to. And under lane 10, this is our control. We just have 5 micrograms of DsrA RNA, ordinary old wild type. Oh, I think I skipped over lane 9 here. What we did was delete the region between the two restriction sites. So we just basically deleted loop 2 with a little bit more deleted as well. So the results of figure 3, what we've done here is each of our mutant DsrAs that we have created. And what we've done is we've measured our activity. So if you look at our wild type, it does a great job of promoting translation of rpos. It does a great job of counteracting that HNS protein, and remember that then allows the capsule genes to be made. And it made a pretty good amount of RNA. Now when we delete stem loop 1, we still have some anti-HNS activity, but we pretty much have no translation of rpos. So that kind of tells us that loop 1 is responsible for this. When we delete loop 2, we still have a little translation of rpos, but we don't have any anti-HNS protein activity. When we replaced our stem loop 3, not much really changed. Remember, we just replaced the natural transcription terminator with a different transcription terminator. When we inserted the in-call restriction site there, so it just changed a little bit of that sequence to the right of stem loop 1. It didn't affect our HNS activity, but it pretty much wiped out our rpos translation. So that's going to go ahead and tell us that this little bit is important, as well as stem loop 1 for that function. Now when we inserted just the ex-haul restriction site, we lost our anti-HNS activity. Didn't lose much of our rpos activity. So that tells us that that little region there, along with stem loop 2, is pretty important for the anti-HNS activity. And when we deleted the region between those two restriction sites, so if you look at that, it deletes quite a bit of important stuff along with stem loop 2. When we did that, our mutant was pretty much useless. So just to summarize what we learned from that figure, anti-HNS activity, we lose it when we delete loop 2. When we delete loop 1, we lose our ability to promote translation of rpos. We lost a little bit of our anti-HNS activity. And of course, the region which was modified by the in-call restriction site is very important in rpos translation function. And both of these activities, the anti-HNS activity, as well as promoting translation of rpos, both of these activities are independent from loop 3. So let's just kind of go over how loop 1 works. Now in our E. coli, rpos, which is just a protein, a stress protein, the translation of that is increased at low temperature or stressful conditions. Now, it's previously known that this is dependent upon our DsrA RNA, and it is also dependent upon the HFQ protein. Now, the requirement for the HFQ protein may be negated by modifying our rpos sequence. And if you're just patient and stick with the presentation, you're going to see this is really interesting. We're going to get to this. But the first loop of DsrA is both necessary and sufficient for rpos translation. So let's kind of take a second to look at what's actually going on. So this is just a nice figure I found online. Here is our DsrA RNA. There's our little HFQ protein. And if you look at our rpos mRNA, you'll see that normally it's closed up and the ribosome is not able to access the little part that it needs to, that Shine-Delgarno sequence. So we're not able to make this into a protein. But if you look over here, the combination of our HFQ protein and our DsrA RNA is actually able to open up this rpos and able to allow the ribosome to start translation. So what our authors predict is that complementarity between the DsrA RNA and the rpos mRNA is very important, and not just the structures. So let's go ahead and get into that. Now, for our next experiment, what do we want to learn? Do mutations which negate the HFQ requirement by disrupting the secondary structure of rpos also bypass the DsrA requirement? So let's go back and figure out what that actually means. Our authors already knew that you could bypass the need for this little protein here by modifying this mRNA so that it's just kind of open all the time. So we're wondering, do those exact same mutations also bypass the need for DsrA? So the way we answer that question, the authors took some mutations that had been previously tested, and they just tested that in bacteria with and without DsrA. Here, the authors don't provide very clear data for this section, and two of the mutations that they referenced, I actually had to track down what those were in a different paper that you can see over here. So, up here I've gone ahead and made that clear for you guys so you didn't have to track that down. So what we're doing here, we're looking at how the rpos mRNA is affected by DsrA. So let's go ahead and play our slideshow. Now in our wild type, if we apply DsrA RNA, we can increase rpos translation about 6 to 9 times. Our mutations SD2 and SD3, the purpose of those is to disrupt the GC pairing at positions 126 and 208. So I think it's either one of these or one of these over here. I'm not exactly sure. I just provided this figure to show you guys our rpos in its closed position. And as you can see, it kind of binds to itself to keep closed, like a little alligator mouth or something. Now, if we add mutation SD2 or SD3, which means, for example, say we just applied mutation SD2, we're going to have a G on top and a G on the bottom in these particular positions. So that's kind of going to disrupt the rpos mRNA's ability to stay closed. So just by changing that one position, we can reduce the dependence on DsrA about 2 times. And if we combine those two mutations, it basically restores the complementarity of those areas. It restores our DsrA dependence to 9 times, just like our wild type. And we've had similar results shown with HFQ dependence in the past. So just to kind of show you here, this is just from figure 4, And here we can see rpos in its closed position. The authors refer to that as the low translation position. And here I've gone ahead and circled DsrA, and this just kind of shows how it can open it and expose that little sequence down here where the ribosome wants to bind to in order to initiate rpos translation. So now we're going to go into some mutations of DsrA. Now all we're changing here is we're flipping around some of the sequences within loop 1, but we're going to preserve the structure of loop 1 when we do so. The intention of this experiment is to kind of determine what's important in loop 1. Is it about structure or is it about sequence? So if you look at our wild type, we have a pretty good amount of rpos translation. But if you take and just kind of flip the stem of the loop over, see how they did that there? We really lose rpos translation. Now, if you just flip the bottom part of the stem, but you leave this little GAC up here and this little CUG as the wild type, we still keep a good amount of rpos translation. So that kind of indicates that this might be a more important part of loop 1. And if you look over here, if we did the opposite of that, we don't really have much rpos translation. So it kind of looks like having this GAC right here is a very important part of loop 1. So let's go a little further into our mutational analysis of loop 1 of our DsrA RNA. So let's go ahead and look at loop 1. This is very highly conserved among species. And something else I'd like to take note, this little underlined sequence, not only is that little bit conserved, but that's also complementary to rpos, which is the mRNA that we want to activate translation of. So let's go on ahead. Just a quick little recap. HFQ and the protein and DsrA, the RNA, work in a pretty similar manner. Now, we think that DsrA likely works by disrupting the secondary structure of rpos RNA, and DsrA uses its first loop to do so. Now, the sequence of DsrA's loop, or at least certain parts of it, are very important to its activity here. And of course, that complementarity to the rpos mRNA is very important. That's what we've learned so far from these experiments. So let's go on into our mutational analysis of loop 1 of DsrA RNA. Now, position number 1 of the DsrA RNA loop 1 is predicted to pair with position number 124 of the rpos mRNA. Now this U, wild type U of 124, is also predicted to pair with the G208. Remember the closed position of the rpos mRNA. And our authors just kind of want to test the importance of this. So let's go ahead and go over what we're going to see in Figure 6b, and then we'll get into Figure 6b. So we're measuring the amount of rpos translation via beta-galactosidase activity. And in this experiment, we're actually testing two variables. We're going to modify the rpos mRNA sequence, and we're also going to modify position number 1 of our DsrA RNA. The reason we're doing this is to understand the importance of that complementarity between rpos and DsrA. Okay, let's go ahead and go over this mutational analysis here in Figure 6b. So if you look up here, all of these bars correspond to the DsrA mutant that we have. Now vector is used as a control. That just means we had our same setup with our pbad promoter and stuff, but there just was no DsrA to be made. Now each of these letters here correspond to what we've shifted loop 1 position 1 to be in our DsrA RNA. Now down here on the bottom, these correspond to our rpos mRNA leader sequence. See how it kind of bonds to itself. So all we've done is looked at these different rpos mutants, and then we've looked at the different ways, different little bases of DsrA interacted with those. So just to see what we learned from this, we've gotten some further evidence to support that there is pairing of DsrA and the rpos leader sequence. Oh, and just to show you, once again, we're measuring the amount of rpos that was translated using that rpos LAC-Z fusion. So, weaker pairings between 124 and 208 are going to relax the requirements for number 1 pairing to number 124. So just to clarify what that means, position number 1 of our DsrA is going to bind around here to position 124 of the rpos. And why do weaker pairings between these two relax that requirement? Probably just because there's less keeping this closed. It's easier to open it back up. Now, complementarity of DsrA number 1 to rpos 124 is always needed for the most efficient performance, as you can see. So let's go ahead and move on. We're going to look at analysis of the unpaired region. That's what the authors titled this section. Now, as we learned in Figure 3, insertion of that in-call restriction site between loops 1 and 2 abolishes translation of rpos. So we know that this little area in here is super important, but let's dig a little deeper. Let's find out why. So, what are we actually changing in this experiment? So let's zoom in a little bit, and right here, I hope you guys can see this, is where we've put that NCO, that's NCOI. I've mispronounced that. So we've inserted that restriction site in here. Here's our wild type sequence, and here is what it is when modified with that restriction site. But as you're going to see, we've once again generated several mutants of DsrA that we're going to be testing. So let's just go ahead. I know there's a lot to unpack here, but we'll go ahead and get into it. So we're looking at sort of the expected interactions between our DsrA and our rpos. So if you look, this little section here, which is the place where we put that restriction site, see there's the wild type sequence of there, and there it is with the restriction site. And this little bit of DsrA is expected to bind to rpos right around here. So just go ahead and note that the wild type DsrA is complementary to rpos in this position. Now over here on loop 1, if we zoom into that, you'll see this little bit here is expected to bind right here to rpos. And it almost makes you wonder if binding here and here and here and here doesn't do something where this sort of bends back and opens it up. That's just a little bit of speculation on my part, so don't quote me on that. Now, authors are performing tests with the modified DsrA as well as some modified rpos mRNAs. And once again, we're going to use that LAC-Z fusion to measure translation of rpos. So let's get into this. Once again, the authors weren't very clear here, so I've gone ahead and labeled their figure myself. Now, on the left, this first group of bars here is just wild-type rpos mRNA, and we're going to test it with numerous mutants of DsrA, and I'll go over those in a moment. Now, over here, we have modified the rpos leader sequence such that it is complementary now to that restriction site in the DsrA. So let's just go on back. So we've modified this little bit right here such that it is complementary to this little bit right here in that version. And of course, the natural wild types of each are complementary to each other. So, just to go over the different types of DsrA mutants, once again we have the vector, no DsrA, and I've gone ahead and added the names of the plasmids, which that just sort of references back to our paper. Now we of course have our wild type. Here we have our DsrA with that restriction site inserted. Now here we have something a little bit unique. We've inserted that restriction site, and then after that we've modified it enzymatically. So basically, I'm not going to go too far into it. What that means is we still have 8 base pairs of pairing compared to 12 base pairs of pairing in the wild type, and that's between our DsrA and our rpos. So that's the difference there. Now, in this last one, this is something we've seen earlier. I believe it was figure 3, but we've just once again deleted that region between the 2 restriction sites. So let's look at the results of that. When we use a wild type rpos, and when that little bit I showed you earlier is complementary, of course it's complementary to the wild type, we get plenty of translation of rpos. Now, over here, we've modified rpos such that its leader sequence is now complementary to that restriction site in DsrA. So, of course, when it's complementary to DsrA, its activity is the best. We also saw some pretty good activity down here. So this one's got a little bit of complementarity. And this is pretty wild. We saw a good amount of activity here in this mutant right here. So that just goes to show you again, loop 2 isn't that important for the rpos translation. And if you look back here at the wild type, we did see a little bit of activity there. Now, just to speculate, again, this is my speculation, I believe the reason why this bar is so much taller than this bar is that we're using a modified rpos. It's probably a lot easier to open back up. In other words, it's not binding to itself very tightly. That is my theory. So, what did we learn from all that? We got further evidence that loop 2 is not involved in the rpos translation function. And furthermore, we've really cemented that complementarity between rpos and DsrA is key for the efficient translation of rpos. So, we're going to move into the discussion portion of the paper, and we're going to wrap up just a little bit. So going back to the title, let's talk about what is an antisense RNA. So this is just my little doodle here. We've got an ordinary single-stranded mRNA floating around. Then an antisense RNA would be complementary to that. So when that gets released, it's going to bind and form a little double-stranded RNA. Those are usually just going to get degraded. But if you look over here at just a simplified drawing of our rpos mRNA, it's almost like it acts as its own antisense RNA. So that's pretty cool. And I would be curious to see if it gets targeted for degradation when it's in this position. But the way we get that title, DsrA is acting as an antisense RNA by opening up our rpos, taking it from this back to this. Just a brief summary of what we've learned today. We've seen that sequences in the first loop, as well as those early in the second loop, those are not needed for HNS silencing to overcome that at all. And we've also seen that the function of HNS silencing is attributed to the second loop up to the third stem loop, and there may possibly, we're not for sure, be some things on the 5' end that are contributing to the HNS silencing. We've seen that the sequences of the first stem loop and a little bit up to the base of the second stem are very important for promoting the translation of rpos. We've seen plenty of evidence that DsrA, of course, can promote the translation of rpos in general. And just something I wanted to note, rpos is also referred to as sigma S, and in last week's papers, we saw sigma S quite a bit. So this is really cool. We've seen a lot of evidence that the complementary binding of loop 1 of DsrA and the rpos leader sequence is very important for its function. We've seen that disrupting the complementarity reduces the efficiency of rpos translation, and we've seen that when we restore the complementary binding between DsrA and rpos, it's going to restore the efficient translation of rpos. Just to quote the authors, going back to the title, DsrA acts as an anti-antisense RNA by relieving inhibitory pairing. So going back into that complementarity, there's about a 20 nucleotide stretch of incomplete complementarity between rpos and DsrA. We've seen a few points where loss of just a single match pretty much wiped out this DsrA activity of promoting translation. We've seen that in positions 1 and 5 of the first stem loop of DsrA. Now we're going to kind of go over some proposed models of how HFQ protein, DsrA, and rpos all interact together. I just pulled up this briefly to show you what HFQ is. It's known to be an RNA chaperone protein. Basically, it helps things like DsrA bind to things like rpos. And it has quite a few functions outside of just this particular case. So with that in mind, we've got some theories about how all these things go together. But let's just go into some background information you're going to need. So at low temperatures, HFQ and DsrA are both required for rpos translation. We also know that more DsrA is made at low temperatures. And remember, when DsrA is overproduced, we get that stem loop 2 function, where we promote the formation of a capsule. That's pretty cool. Now, at higher temperatures, we still see dependence on the HFQ protein, but our requirement for DsrA is relaxed at high temperatures. So, what's going on? Is HFQ opening up the mRNA all on its own? Well, possible Model 1 that the authors proposed is that environmental signals are just acting on HFQ or on the rpos leader sequence directly at a higher temp. But the model that I personally agree with, and I would guess that the authors probably support this model as well, we're thinking maybe there are other DsRA like molecules out there. So at high temperatures, instead of DsRA being here, maybe there's some other RNA that we don't even know about yet. So HFQ, of course, in that model would just be acting as an RNA chaperone, which is something that it's been shown to do. This is kind of interesting. OxyS, it's an oxidative stress response, and it's also known to act on rpos, down regulating its translation. And the way it does that is it actually acts as a competitive inhibitor of the HFQ protein. So that's pretty cool. And like DsRA, OxyS has more than one target in the cell. So that just goes to show you there's probably a lot more of these little RNAs that are regulating things. Right, so let's briefly go over our materials and methods. Most of our bacterial strains, I think all of them actually, had their natural lac operons deleted. And our rpos lac fusions, and they didn't specifically mention the other lac fusions in the paper, so I'll assume it was done the same way. But our rpos lac fusions specifically were inserted in the trp operon, and the way they selected for them was by including a kanamycin resistance gene, just to knock out all the ones where the lac fusion didn't take. I'm sure you guys are familiar with that kind of method. We use some strains with just their ordinary natural DsRA. And then we described this earlier, but our DsRA plasmid was under PBAG control in the strains where the natural DsRA was deleted. Then of course we had some versions of that plasmid with the DsRA left out, as we mentioned earlier. A lot of the constructions of the plasmids and the transformations they described in this paper, I'll just be honest, it's very much above my understanding. I've never worked in a lab and done those things myself, so that may be part of it. Our northern blots, they used a QIAGEN kit that you can purchase to isolate the RNA, and they added 3 micrograms of RNA plus some buffer solution so that it added up to, I think, 20 micrograms or microliters total volume that they put into each lane. Our beta-galactosidase assays, this was described in a different paper, but I went ahead and looked that up, and it was pretty simple. They grew the strains in microtiter plates, and that's just like those little well trays you've probably seen before. Maybe you've micropipetted things into that. They measured the absorption as a function of time, and I showed that in purple a little earlier. They measured the specific activity. And the authors noted a few times that their specific activity is about 25 times less than Miller units, which I guess is just another unit of measurement people use in this case. The machine they used to measure absorption, they used the SpectraMax 250. And you can pick one of these up off the used market for about $7,000. The way we determined RNA half-life, they used DDS92 cells. I've searched through the whole internet and I was not able to find what that is. So I'm assuming it's just something specific to their lab, and I'm just going to assume that it's regular E. coli, but don't quote me on that, okay? So they grew it in LB medium at 32 Celsius. During the exponential growth phase, they added this little chemical. It's a transcription inhibitor. They isolated the RNA as I've already described, but the difference is they used 5 micrograms of RNA per lane instead of 3 micrograms of RNA per lane. So that's all I've got for you guys today. I really hope you enjoyed it, and I really hoped I was able to clarify the presentation and just make it easier to understand for you guys.